There we go. All right, Brooke.
Excellent. All right, it looks like people are coming in. So welcome everyone. It's just about 7 o'clock. So I will wait to do the introduction, but I do have a couple more announcements. I sent an email out today to folks. So if you didn't check your email yet, there's a couple more items this week happening. One is that on the first, so that's Wednesday, I believe, our colony loss census is opening. So if you've been tracking your losses and diseases and pests from last year, that will open. And so while it's fresh on your mind, you can fill that out. And then on Friday, we have a lunch and learn with the New England and plus New York apiary inspectors. And it sounds like I might be with some apiary inspectors in New York on Friday. We have the from noon to 1:30, a roundtable. We share what's going on in the field and then take questions from beekeepers. And it's kind of like a really fun talk radio show. So join in on that if you have time and while you're eating your lunch. It's a pretty casual event and fun time to share. So let's see. I think folks are still maybe trickling in. I can't really see if there's like people flooding in, but I think we could get started if that works for you, David. I'm gonna pull up my very short introduction here, but I'm Brooke Decker, if I didn't say that already, the apiary inspector here in Vermont, and most of you know me, and many of you probably know David as well, but he is the Director of Research and Education at Betterbee in Greenwich, New York. And David has done extensive research on honeybees and Varroa, which he's going to be sharing a lot about tonight. And I know David is a great collaborator, both locally and on on a national and industry level. I think David has been at every conference I've ever been to, and many of the working groups. So I get to see David a lot. So we're really honored to have him here to share with you all and me too. I always learn something from David. There's a lot to learn about Varroa. It's a constant threat, and so very honored to have David here. And one little side note about some housekeeping. We'll keep you muted. I think you're muted when you come in. So stay muted during the presentation and then hold your questions till the end. And we are going to use the chat box, not the question and answer section. So if you have questions, we'll save them for the end, but if you want to write them in the chat box, we'll go through them at the end. And then we'll probably check the Q&A section if something ends up in there, but just stick with the chat. And without further ado, I would like to welcome David.
Great. Well, thank you so much, Brooke. It is a great pleasure to be here. And as I look through the names of folks here, I know that I know some of you and don't know others. It is always my pleasure to talk about Varroa mites because it's something that I think is the linchpin in other beekeepers keeping their bees alive and healthy. And so without further ado, I'm going to be very careful. The end event button is right next to the share screen button. So I'm going to click very carefully over to here so that I can share my slides. And then I think I should be able to pop this right up and we should be off to the races. So what we're doing here, the hope, I believe, at least of today, is that we are going to really go through an overview of Varroa mite biology, talk a little bit about the history and the biology and the physiology and the development of these mites, and then talk about management options that are available to beekeepers like you. And so for some of you, this will be a review. For some of you, this might be new information. And then at the end, I'm very happy to answer questions, as Brooke just said. So just we'll run through sort of the presentation, and then there's time for discussion afterwards. So, and I have to make sure I can advance my slides. There we go. So I won't belabor the point talking about myself too much, but I got my PhD at Cornell working with Tom Seeley, and my focus there was on bee behavior, Varroa mite behavior, and the behavioral interactions between bees and Varroa. So the behaviors of the mites, but also the behaviors of the bees towards the mites. And in my role now at Betterbee, I really have been able to continue a lot of that research, but also get myself involved, as Brooke said, in a whole lot of different working groups and other activities around the world, working with bees. And I also, teach classes at Betterbee and work on product development and things like that. So the learning objectives that I've set for this presentation are, I want you by the end of this to be able to explain why Varroa mites need to be managed in your apiary. I want you to understand various chemical methods that are currently available to treat your mites, and that's going to include some brand new stuff. I want to at least summarize some of the non-chemical mite management techniques that you might be using. And I want to talk about this mite management mindset, which I think is the foundation of keeping your bees alive. Regardless of what interventions or non-interventions you are choosing to use, whatever management you're using that's keeping your mites under control, this mindset of when, why, and how we need to be thinking about Varroa, I think is really important to succeeding. So this is a Varroa mite. This is what we're here to talk about. Varroa destructor, if you didn't know anything about them, the name would sort of give it away, are these devastating parasitic mites of honeybees. And the fact that they are mites, that they are arachnids, they have 8 legs instead of 6 like our bees, already gives us a little bit of an advantage in fighting them. Because obviously, if I can find a chemical that kills arachnids and doesn't kill insects, already I've maybe got an angle that I can use to try to get these things under control. But the problem with these mites, the reason that they're so devastating, is that they underwent a very recent host jump from the original species of bee that they lived on, Apis cerana, the eastern or Asian honeybee, onto Apis mellifera, the bees we keep, the Western or European honeybee. And so our bees basically found themselves launched into a battle against this parasite that had been fighting a war against Apis cerana for, who knows how long as the mites and the bees had been interacting. So Apis cerana has some tricks up their sleeve. The Varroa mites had some counters to those tricks that were up their own sleeves. And our bees were just naive and suddenly found themselves suffering terribly when these mites jumped onto them. So the summary essentially now is that if you've got honeybees, you've got Varroa mites. Betterbee sells nukes and packages to new beekeepers all the time, and we teach them about mites. And when they pick up their bees, they often say, "Hey, you don't have any Varroa mites on these, right? Do you guarantee that these are Varroa-free bees?" And I tell them, not only do I not guarantee that they're Varroa-free, I guarantee that every colony you get from us has got at least one mite. And all it takes is one, because they can simply reproduce and reproduce and reproduce and reproduce and grow exponentially. So you need to begin with those bees with an understanding that Varroa mites are part of the mix. They alternate between 2 different behavioral modes in their lives. What we call the reproductive mode, where the mites are entering brood cells, they feed on the hemolymph, essentially the blood of those pupating and developing bees, and then they lay eggs and their offspring, grow and develop from there. And then another mode, which is sometimes called the phoretic stage and sometimes called the dispersal stage. And neither of those is exactly right, if we want to get quibbly and technical about it. But both terms are often used. You'll probably catch me saying phoretic more often than dispersal, even though that technically isn't the right word for it. But this is the stage where these mites are riding around on adult bees. More often than not, that's going to be the nurse bees in the hive, although we know that they will feed on other bees. And they feed on the fat bodies, these organs inside of these bees' bodies that are critically important to overwinter survival, to their ability to function as good nurse bees, and in order to live long, healthy bee lives. And it's very hard to nail this down, but we know that most mites typically go through a couple of these reproductive cycles in their lives. They'll be reproductive, and then phoretic, and then reproductive, and then phoretic, and then, at some point, they give up the ghost and keel over. So in that reproductive mode, what these mites are actually doing is very intricate. And this is a key lesson that I learned in my study of Varroa, that you can never underestimate a parasite like this. Because even though they're little and they're gross and we don't like them and we wish they were gone, they're still very sophisticated animals. They're very good at doing what they do. And unfortunately, what they do is parasitize honeybees. So here we can see on the left, this foundress mother varroa mite is going to run down into a cell just before it gets capped. And you can see her down at the bottom of that cell, drawn in, sort of snorkeling inside the last remaining bites of brood food that larva is going to eat. Then, as that prepupa stands up and begins spinning its cocoon, the mite is going to crawl up and start feeding on it. And so as that bee pupates, the mite is going to be laying a series of eggs. The first egg that she lays is male. All of the subsequent eggs that she lays are female. And that young male and those young females are going to also feed on the bee. And then eventually they are going to rendezvous inside of the hive and incestuously mate with one another. And so the end result is that we've got a whole bunch of pregnant female mites, including their mother, climbing out or riding on the bee as it climbs out of the cell. The male mite is left behind. Any immature female mites are left behind. And now we've got another generation of pregnant varroa mites that maybe after a few slurps of nutrients out of a couple of adult bees is going to be ready to go in and start reproducing again. And so this is why just one mite in a package or a nuke isn't going to lead to a population collapse due to inbreeding. Basically, inbreeding is built into the system here, and so they don't suffer any consequence from mating with their sisters or their cousins or what have you. In the phoretic mode or the dispersal mode, you've got the mites, as I said, riding around on the bees. And this is a photo that I took during my graduate work, and it's one of my favorite photos I've ever taken because I think it teaches an important lesson. I took this photo of a colony that was absolutely riddled with Varroa. I had bred as many Varroa into the colony as I possibly could in an attempt to kill them. And what I want you to notice is that I took this photo through the glass wall of an observation hive. All of these red circles are showing you not just one, but often more than one Varroa mites on the undersides of these bees' abdomens. But what I want you to notice is all of the other bees who are crawling around on the comb, the bees whose backs and wings we can see. Those bees probably have Varroa mites on them, but we can't see them because these mites aren't just blindly meandering around on the surfaces of the bees. They are deliberately and pointedly positioning themselves on the undersides of the abdomen where they have access to food from the bee's body and where the bee has a hard time grooming herself. And so if you're going into your hives and you're not picking your bees up by the wings and flipping them over and looking at the bellies of all of the workers, you're not going to be able to visually assess the infestation level of Varroa. And as I said, as they're riding on those bees, they're going to be feeding on the fat bodies of the bees. So this is a freeze fracture image that was created with the help of some of the resources at the USDA ARS. And it shows a mite and the portion of a bee's body that have been frozen in liquid nitrogen and then basically cracked right down the side. And so we can see where this mite is sticking its mouth parts into the bee and is sucking out this fat body tissue, which is now going into the mite's digestive tract. If it wasn't bad enough that they drained nutrients and energy and longevity out of our bees, they also, very importantly, transmit viruses. They're vectors of virus transmission. So there's a whole bunch of known honeybee viruses that are listed over here on the left. There's also, obviously, a bunch of unknown honeybee viruses, things that we haven't been able to characterize and categorize yet. But we know that these mites, as they feed on these bees, transmit viruses, things like deformed wing virus, which at high levels can cause the wing deformities that you can see on the right. And so these deformed wing virus and other viruses that build up inside of the bees' bodies get essentially transmitted and retransmitted and amplified and multiplied as these mites feed from bee to bee to bee to bee, adult to larva, adult to larva, adult to larva. It's not just deformed wing virus that you need to worry about. There are other viruses that the mites can also transmit. So these are images of a colony that have high levels of acute bee paralysis virus, which is characterized by dead, dying, and discolored and contorted brood, which we can see in photos A and B. And in photo C, this sort of characteristic dark, black, almost greasy looking bee who's probably going to have a hard time moving smoothly around the hive because she's got very high levels of this paralysis virus. So it's more than just wing deformities that can be indicative of viruses that can themselves be indicative of high levels of mites. So this graph gives a very basic overview of sort of the way we imagine the bee population and the mite population changing and growing over the course of a season. So at the bottom of the graph, you've got January running all the way along to December, and we can see that at the beginning of the year, the yellow line, the bee population, starts relatively low. It increases rapidly in spring. It reaches a peak, and then it starts to drop off. And then over the course of the winter, due to attrition, we've got fewer and fewer bees in the hive until, unsurprisingly, we start off exactly where we were at the beginning of the previous year. And so the bees simply run in that cycle. The mite population, though, starts very low in this graph, and we can see that it grows exponentially, lagged a little bit behind the bee population. But essentially, every time those bees have brood in the colony, it's an opportunity for the mites to reproduce and the mite population to increase. It also hits a peak. They also have some mites that die off over time as brood rearing slows down and stops. But by the end of the season, we've got a lot more mites than we started with. In addition to all of that, if we look at the area underneath that red line and we imagine what it represents, we can, think about our high school calculus, and we can think, hey, you know what? I guess the area under that red line represents something. And in a way, because that represents the increase over time of the mite population, and we know that in order for the mites to reproduce, they have to feed on the bees, that shows sort of a rough estimate of the overall mite feeding activity that's happening inside of the hive. That means that it also correlates to the overall accumulation of viruses inside of the hive. And so when we have a high level of Varroa, it means that we're going to have a high level of Varroa feeding and therefore higher levels of virus transmission. The take home here is that you need to be thinking not just about getting your mite levels low at one point in the year, you need to think about keeping your mite levels under control throughout the year. Because a huge spike from May through September, and then you treating and killing your mites and knocking them down to zero, does not eliminate all of the harm that those mites have done. In fact, the levels of virus could be so high after you do that September treatment that your bees could die, and it could be the fault of the parasites and the viruses they transmitted, even if you've killed every single parasite in the hive. So if that wasn't bad enough, these mites are also, as I said, very good at what they do, which is being parasites. And they're very good at transmitting themselves from one hive to another. There's a few different ways that can happen. The Varroa can't fly. They, in fact, don't even have eyes. So they'd have a hard time if they crawled out of the hive and started looking around for other bees. But we know that they can transmit themselves by clinging on to drifting bees and having those bees get disoriented and go into the wrong neighboring hive. This happens more often within an apiary than between apiaries, but we know that bees will drift long distances sometimes. There's also an opportunity to transmit during robbing. If bees from a healthy hive fly over and they rob from a colony that is sick with Varroa, they might be able to steal some honey, but they might also pick up a Varroa mite on the way home. And so both of these are mechanisms that allow sick colonies to get their mites into healthy colonies and spread the mites across the landscape. It means even if you killed all of the mites in your hive through some miraculous combination of management and luck, you would still wind up probably picking up mites from other colonies relatively quickly. It also means that as beekeepers, we don't want to be bad neighbors. So if you lived in a neighborhood and this was the neighbor next door, it's the kind of thing that would make you wish for a homeowners association, some way that you could make them stop keeping all this garbage around. And in the same way as beekeepers, if we're not keeping our mite levels under control, that can impact the mite levels of beekeepers nearby. So the best way to keep mites under control or to know that you're doing so is to actually monitor and count the mites in your hive. The best way to do that is an alcohol wash or a soapy water wash. of the phoretic mites, the dispersal stage mites riding around on the adult bees, where we take a sample of bees and then we knock the mites off. The thing to keep in mind, though, is you're only ever measuring the phoretic population of mites. Unless you're going and opening up brood cells, you don't actually know what percentage of the mites are underneath those caps. And so this is still going to be only an estimate, a metric, an index of your mite population, but it's not an absolute direct measure of how many mites there are unless the colony is completely broodless. So just very quickly, the alcohol wash is so easy to do that I can demonstrate it right here in a few photos. This is our head beekeeper, Anne, who has shaken a frame out of the brood nest into this white plastic tub. She checked it beforehand to make sure the queen wasn't there, because we don't want to subject her to this. And she's giving one last check just to make sure that she can't see a color mark from a queen who she had missed earlier. So she shakes the bees down, she knocks the bees off into the corner, and she takes a measuring cup and measures out a half cup of bees. No need to level it with a butter knife, close is good enough. We take that half cup of bees, which we know is going to correspond to 300 adult workers, and we put them into our prepared Varroa monitoring jar with some rubbing alcohol or an alcohol-based windshield wiper fluid inside. We put the lid on, we say, sorry bees, we swirl it around and we kill all of the bees, and we dislodge all of the mites from them. And then you can discard the bees in the grass and count how many Varroa mites have been dislodged from that sample of 300 bees. So looking here, each of those little dots is a Varroa mite, and I can say this is a colony that probably needs some kind of intervention because that mite level is probably going to be dangerously high for that colony. The other method that you can use and that absolutely can give good, useful data is using a sticky board underneath a screened bottom board. So the idea here is that all of the hive detritus, including dead mites, are going to fall down through that screen. They're going to land on this board that you've covered in oil, and then you can measure how many mites fall in 24 hours, 48 hours, 72 hours, 1, 2, or 3 days, whatever you choose. It's important to understand that this isn't as accurate as other methods, right? If the colony is doing something weird up above, or if there's something on the bottom board that's keeping some of the mites from falling through, you can have some skew to your data one way or the other. But it's still useful. And if you as a beekeeper aren't going to be doing alcohol washes, if you know in your bones that you're not going to get around to doing it, this is still going to give you some information about your mite level. So it's not useless by any means. When is it recommended that you treat for mites? Well, that question really comes down to the principle of IPM, or integrated pest management. The idea is you don't want to be applying pesticides willy-nilly to your hives for no reason, but you do want to use these products or do these interventions when you know that knocking the mite level down is going to be important for the health of your colony. So you set a threshold and you say when the mite level gets above that threshold, that's going to be when we do an intervention. These are the current recommendations from the Honeybee Health Coalition. I'm actually on the Honeybee Health Coalition committee that is revising the Varroa Management Guide, and that includes this table. This table. And I can tell you that some of these numbers are going down. The recommendations are becoming less permissive of higher mite levels. But the recommendation is approximately in winter, you treat when the mite level gets above 1%. In the spring, summer, and fall, somewhere between 2 and 3%. It was the recommendation before. If you're using a sticky board, then generally just whenever you have more than 9 mites falling per day, that's the rule of thumb that tells you, this is getting to be problematic. I should probably take some action. But if all of this is a little too much, the rule of thumb that many beekeepers have been using and using with success is that they treat their colonies whenever they get to or above 2% infestation. So that's 2 mites per 100 bees, but since the sample is of 300 bees, this means that if you get 6 or more mites, you're going to do a treatment. If you get 5, 4, 3, 2, 1, or 0 mites, it's not time to treat yet, but you're going to monitor again, and you'll treat when the level gets higher. So I now want to give, as best I can, a summary of all of the different control mechanisms that are available to beekeepers to keep their mite levels low. And so I'm going to start with chemical control because I have to start somewhere, but I am going to run through other options as well. So there are a lot of different registered miticides that beekeepers can buy and use inside of their hives to control Varroa. I've listed, I think, all of the ones that are commercially available right now up here. But at first you might think, hey, this is great. There's all sorts of products, fantastic. I can grab any of these. But then you might, stroke your beard and think, why would they keep making new miticides if we already have so many of them? And the answer is that there are some problems here. The first two miticides in that list, fluvalinate and coumaphos, or rather the products are apistan and checkmite, the mites very rapidly evolved resistance to those chemicals, and so we can't use them anymore. They're no longer valuable for mite management. The next synthetic pesticide in that list, amitraz, we know that the mites are now evolving resistance to, and we're seeing higher and higher levels of amitraz resistance, particularly in some commercial beekeeping operations. But it can happen to any beekeeper because these mites spread so well across the landscape. So suddenly this long list starts to become shorter. But at the same time, new products are coming out all the time that maybe give us some more options for managing Varroa. So before I get started, I want to emphasize that the label is the law. This is the mantra for all pesticides regulated by the EPA. Whatever is printed on that product label that is sitting there in front of you that you're holding is the law that governs your use of that product, in your hives. And so if I say anything right now, or if anything in my slides doesn't correspond to the label, if I'm mistaken in any way, or if I misspeak, understand that you are governed by the label that is printed on the product that is sitting in front of you. So you should always read it. So very briefly, I will just summarize these different products and some of the pros and cons and limitations of them. So the first is the active ingredient is amitraz. So the products that are available are Apivar and Apivar 2.0. Apivar is no longer being made, but there may be a few packages of it kicking around in your shed somewhere. But Apivar 2.0 is the sort of new and improved, very similar version of this product. It's the synthetic pesticide impregnated inside a plastic strip. It isn't safe to use with your honey supers in place because it's a synthetic pesticide. It needs to make contact with the mites, and it only kills the phoretic mites in the hive. And so you put this strip in, but a lot of the mites at that time are going to be protected underneath the brood caps. But because the strips are in the hive for so long, typically 6 to 10 weeks for a treatment, the mites that were protected earlier are going to come out and wind up getting exposed. And the temperature range is pretty permissive for this stuff. There's another product based on amitraz called Amiflex 2.0. So the idea here is that you basically are going to use this caulk gun-like applicator to put a little strip of this, or a couple little strips of this, inside of the hive. It's a flash treatment with amitraz. Still not safe to use with your supers in place, but it has a slightly different mode of action. And so there may be some advantage to using this under certain circumstances. Now, a lot of you may have heard about Amiflex as a product which required a commercial pesticide applicator's license in order to use it. But that restriction has been removed by the EPA, and the company that makes it is currently just rounding up the last few states and getting them to approve the new version that doesn't require that commercial pesticide applicator's license. So I've got a photo here of the original product, Amiflex, and then if you pay attention, that's a photo of the new product, Amiflex 2.0. Did you see that? So if we go back and forth, basically they've just added the number 2 to the package. That's it. It's the exact same product made in the exact same way with the exact same rules. It even costs the same amount. It's just that it doesn't have this extra restriction on it that made it very difficult for anyone who didn't have an applicator's license to buy it. The next active ingredient is thymol, and there are 2 products that use thymol that are available to beekeepers. One of them is Apiguard, which is a thymol gel that is put on top of the hive. In this case, it comes in these little metal trays, sort of look like a wet cat food tray or something. You can peel that foil off, put it on top of the hive, and the bees go up into it and they start hauling it out and trying to move it around and get rid of it, and the mites wind up getting killed. The other product is Apolife Var, and I put an asterisk here because that actually has more active ingredients than just thymol. It's a mixture of essential oils, but it's soaked into these wafers, these sort of briquettes that you put on top of your top brood box, and then that creates a vapor that goes down through the hive. Both of these need to be reapplied in order to catch the mites that were protected under the brood caps before, and neither of them can be used with supers in place because it's going to stink up your honey. It'll start smelling like mint or like thyme because thymol is derived from the essential oil of the herb thyme. Formic acid is available in the form of the product Formic Pro. This is pretty straightforward. It is these strips, these polysaccharide formic acid strips wrapped in this wicking paper material. You put it in between the brood boxes of your hive. You can do either one strip or two strips to treat a hive. It creates a vapor, and that vapor kills the mites, including penetrating the brood caps and killing the mites that are actively reproducing. That's a cool advantage of this product. It's also safe to use with your supers. The disadvantage is that its temperature range is not very permissive. When the temperature gets below 50 degrees Fahrenheit and the colony goes into cluster, or above 85 degrees Fahrenheit and the colony starts trying to actively cool themselves, they aren't able to disperse the formic acid vapors as predictably and efficiently, and so the colony can wind up suffering as a result, and you either will get very poor mite control or you'll get good mite control. Lots of mites are killed, but you'll also start killing a lot more brood and bees than you want. Another product based on hops beta acids is called HopGuard 3. This is the third incarnation of this product. It's safe to use with honey supers in place. It only kills the phoretic mites, so you need to reapply it if you have capped brood in the colony. And it is also quite expensive. I believe it is the most expensive product per hive treatment, or at least it gives some of the other expensive ones a run for its money. And so it's a little bit gloopy to use. A lot of folks don't enjoy messing around with this stuff, but it certainly can kill Varroa mites. Now, a new product on the market is called Varroa, and Varroa is based on the active ingredient of Vadiscana. This is the very first product to be released for honeybees that is based on RNAi technology. This is a genetic technology, and RNAi stands for RNA interference. And the basic idea is, if you look over at the right, there won't be a quiz afterwards, so don't worry if you don't understand all of this. But the basic idea is that your Varroa mites, just like your own body, all of these cells are taking their DNA and they are making taking little sections of that DNA and then using that as the template to make proteins. Well, if you have a molecule that can go in and sneak in and interrupt that process, that can mess up, interfere with that RNA, then the organism isn't going to be able to make the correct protein. So in this case, the protein that they've targeted is a protein that Varroa mites use to make eggshells. And it means that they're essentially producing what the company has sometimes glibly called birth control for mites. It doesn't actually kill the Varroa, but the mites that get exposed to this stuff absorb it, it goes into their body, and it makes it so that they can't make proper eggshells. How do they get exposed? Well, remember that slide where I showed you the Varroa mite crawling down and hiding in the brood food right before that cell got capped and that larva stood up and pupated? Well, the product is mixed into sugar syrup. And this genetic material is put into the sugar syrup, you feed it to your bees, and then that gets incorporated into the brood food. So while that mite is down there snorkeling, waiting for that bee to get ready to pupate, it's absorbing these pieces of genetic material, which are then interfering with their own ability to lay eggs. So this is a brand new product. It's most folks have not had a chance to try it, although there are some studies that have shown very promising results. But you need to be careful about exactly when you feed it to your bees, because you don't want them to eat all of the genetic material because they're hungry, nor do you want it to get so diluted in nectar from a big honey flow that it doesn't get into the brood food. So the last but not least class of products are all based on the active ingredient oxalic acid. Oxalic acid is a natural compound that is found in honey plants. It's found in honey. It's found inside of beehives, even if no treatments are being used. It's found in sumac flowers and the nectar of oxalis wood sorrel flowers. And there's a lot of different products that use oxalic acid to control Varroa. What's interesting is that oxalic acid, like formic acid, which I've shown you images of both of them over here on the right, oxalic is basically just two formic acid molecules glued together at the carbon, but they're chemically very similar to one another. Both of these products kill Varroa mites. We actually don't know exactly how they do that, which is sort of weird to admit. It may be that they're acidifying the mites. It could be that they're damaging their chemical sensing hairs or their feet and the foot pads that they use to navigate the bees. We genuinely don't know exactly how they do it, but it works. So how can you use this chemical to kill Varroa mites? One method is through vaporization. So this is, there are two products, apibioxyl and EasyOx. Both of these can be put into a heat vaporizer. These are battery-powered vaporizers being demonstrated here. You need to wear quite a lot of safety gear to keep yourself from breathing in the cloud of oxalic acid that is going to be produced by that vaporizer, and then is going to hopefully not float out into the air or into your lungs or your face, but go into the hive and recrystallize, forming a cloud of microcrystals that's then going to cover every surface of the hive, including the mites. You do not want to go to the store and buy wood bleach, which is also oxalic acid, and use that, because doing that is a violation of state and federal law and could expose you to some serious liability. So you want to use the products that are actually tested and manufactured to guaranteed to be safe for bees. And if you do that, it is safe and legal to use this with your honey supers in place. Randy Oliver has been doing tests for quite a long time on different ways to apply oxalic acid, and you Vermont beekeepers had a brief opportunity where you were permitted to make your own oxalic acid strips that could go into the hive and provide an extended release of that oxalic acid. Well, back and forth between the EPA and state government led to that permission going away, but there is luckily now a product that is available that you can use that has much the same effect. And so there is a product right now called Viroxan, which is available, which is oxalic acid and glycerin soaked into fiberboard strips that you put down over the tops of the frames of your hive, and that leads the bees and therefore the mites getting slowly exposed to this over time. There's also another product, an older product called Uncapping. Viroxan is really just a knockoff of Uncapping, but Viroxan got registered in the U.S. before Uncapping did, although they are looking into the process of getting themselves registered so they can sell into the US as well. Safe to use with honey supers in place, although with some little caveats about the positioning of it, and a very cool product that a number of beekeepers have found useful. However, a lot of research has gone into the value of these extended release oxalic acid pads, and the basic summary of that research is the following. Extended release oxalic acid strips seem to be pretty good at keeping low mite levels low, slowing the rate of mite population growth, or keeping a low level that is under control for longer. They are not terribly good at knocking high mite levels down. So once your mite level gets to an infestation, maybe above that 2% threshold, throwing in these oxalic acid strips doesn't seem like it's going to do a very good job of knocking down very high mite levels. The same is true, at least in the research I've been able to take a look at, for Varroa. Varroa seems like it's good at keeping low mite levels low. It's not terrific at taking a really high problematic mite level and then crashing it down as close to zero as possible. So there's another way that you can use oxalic acid, which is mixing up those powders with sugar syrup and then dribbling that onto your hives. This has been permitted in the United States since 2015. And so it's a method that a number of beekeepers have used, mixing the OA into this OA syrup and then putting it on the hives. That is also approved for use with honey supers in place, and it only kills the phoretic mites. So if you've got brood, you would either need to reapply it or just understand that you're not going to be killing all of the mites in the hive or even exposing all of the mites in the hive to the treatment. There's a new product which has just hit the market called apibioxyl RTU. That is approved by the EPA and it's approved by most states. There's just a few that have been delayed in getting the approvals through. But this is oxalic acid that has been mixed with inert ingredients that do not include sugar. So the problem with the OA dribble treatment, the sugar syrup one, is that bees, understandably, have a temptation to eat it because it is sugar syrup mixed in with the medicine. And so obviously it doesn't last in the hive very long if the bees are in there eating it and cleaning it up and moving it around. So this is ingredients like glycerin and water that are mixed with the OA, and it produces this not very tempting fluid for the bees to eat, and it seems to increase the residual. It's also at a higher concentration of oxalic acid than the stuff that you can make in your own sugar syrup dribble. And the dosing of it is a little bit more straightforward too. There isn't a maximum dose per hive that you have to add up to. You just have to put 5 milliliters of this stuff dribbled onto the heads of your bees for every seam of bees, every space in between the frames that is occupied by bees. Also safe to use with honey supers in place. So, oh, I do. Yeah, I have just a little video here showing this stuff because it is pretty cool. This is what I'm probably most excited to use in my apiary this year. And the idea is essentially that you're gonna dump this ready to mix, you're ready to use, no mixing, no math, no calculation required material into a syringe or into one of these doser bottles. So in this case, you're gonna dose that up to, let's say, 50 milliliters, and then I can dribble this, just trickle it down out of that dosing bottle directly onto the heads of the bees in between the frames. And as long as I get through that 50 milliliters, by the time I've gone through 10 frame spaces, I figure I've got 5 milliliters per seam of bees. A real advantage of this stuff is that you don't need to wear the same safety gear. You do still need to wear eye protection. They encourage you to wear a mask like an N95 mask or something like that. You do want to wear gloves, but the risk of exposure to this stuff is much lower than the risk that you are exposed to with the vaporizer. As long as you don't squirt the stuff up your nose, in your eyes, or into your mouth, the likelihood of being seriously harmed by it is pretty low. So all of those chemical controls that I've now summarized exist. They're all available. What's the value of them? What should one do with them? Which is the best of these that I should be focused on in my own mite management? Well, I think that's fundamentally the wrong question. Because really, each of these products is a weapon in your arsenal in the war against Varroa mites. They're all important tools for you to understand, which is why I've just went through every one of them. Because different compounds may not be effective, right? Maybe your mites are resistant to them, and so some of those older products are no longer useful. Some of these products are great, but if it's 95 degrees Fahrenheit, you can't use something like Formic Pro, because you're not allowed to use it above 85. Some of them aren't very effective if you've got brood. Oxalic acid products, HopGuard products just aren't that good when you've got lots and lots of brood to protect the mites. And some of them aren't suitable if you've got honey supers in place, right? The thymol products or the synthetic pesticides. So we can't just keep waiting for one silver bullet. There's nothing that makes me more uncomfortable than a beekeeper who says, oh, I'm a Formic Pro guy. Formic Pro is the only way I control my mites, or all I use is oxalic vapor because that's the thing for me. I think it's important to recognize that using multiple products is best management practice, and it also is going to make you more successful in what you're trying to do. If we just use one product, if you overuse just one treatment, what you're doing is you're basically selecting for mites that are resistant to that product. And so we know that fluvalinate, apistan strips, Coumaphos, check mite strips, those products were the only thing those beekeepers were using over and over and over. And so there's no surprise that the few mites that could survive started spreading, and we wound up with resistant mites. Amitraz is still an effective product as long as your mites aren't resistant. And if we rotate between Amitraz products and other treatments, we know that Amitraz regains its effectiveness. The mite resistance goes away, and so we can then redeploy this tool. And so it is important that we rotate, in part because it's going to keep more of these weapons available to fight the mites. Now, that's chemical mite control. But what about non-chemical mite control? What if I don't want to use those products, or I don't want to use as much of them or as many of them? Well, there are a number of different methods for non-chemical mite control, and there's some that you might hear that I have written up here as less helpful. some folks look at the screened bottom board. We know the mites will sometimes fall through onto that sticky board underneath. And so they'll say, hey, if I just take the sticky board out, those mites are going to fall, they're going to die, they're going to get eaten by ants in the dirt, and now my bees are going to be safe. Well, yes, some mites will fall, but generally the mites that are falling onto that board are mites that are at or near the end of their lifespan anyway. So it's not like it's going to treat the mites inside of the hive. It's just going to slightly increase the rate at which they die. Sugar dusting is a method that you'll hear folks talk about sometimes, taking large amounts of powdered sugar and dumping that over all of the bees inside of the hive to dislodge the mites. Yes, we know that that will knock a few mites off of the bees, but by the end of the day, the amount of powdered sugar that you have to dump onto your bees in order to keep the mites under control is so high that you might as well just be putting that powdered sugar in water and then eating that instead of your honey. So it's not the most efficient way to do things. And heat is also a really tempting solution for Varroa. There have been a whole lot of different products that have been developed over time that have been based on this true fact that Varroa mites are more sensitive to high levels of heat than the brood that they are reproducing on. So if you can heat up the colony, you can kill the mites without killing the brood. And that's true if you can make it all work. The problem is that bees are really good at thermoregulating their own colonies. And so often these products do their absolute best to try to heat up the hive, and then the bees do everything in their power to keep it from getting so hot that the mites are well controlled. So this has promise. There's still ongoing work trying to develop a good heat treatment product, but at this point, I have yet to see one that really seems like a slam dunk. There are two non-chemical mite control techniques or management tools that are more useful. And that's drone comb trapping, which I'll describe, and also different methods that involve interrupting brood rearing inside of your colony. And I'll talk about different ways that you can do that as well. So drone comb trapping really hacks into the biology of the mites and takes advantage of what they're trying to do. When Varroa reproduce, we know that more daughter mites are reproduced on drone bees than are on workers. And that's simply because the drones are capped for longer. So that mother mite in there laying egg after egg after egg can get more of those female, daughters matured and mated with their brother before the bee crawls out of the cell. Because of that, Varroa, as I said, are very good at their job. And so they are clever enough to go preferentially into drone cells. That's where we have an advantage. Drone bees come out of drone comb, larger drone-sized cells. Varroa mites, as we said, prefer feeding on drone brood. So if you have a frame, like one of these green plastic frames, that produces that has a larger foundation size and causes the bees to make lots of drone comb, you can put that into the hive, have the bees fill that up with drone brood, and as those drone brood are capped, all of the mites are going to be eagerly running in, rubbing their little forelegs together, saying, great, this will be a perfect opportunity to have lots of daughters. Well, once those cells get capped, if you pull it out at the right time and put it into the freezer or just scrape out all of that drone brood, you are able to then kill all the drones, but also kill a whole lot of those mites. The issue, of course, is that if your timing isn't perfect, if you sprain your ankle and you can't go out, or if it's too rainy and you don't want to bother the bees and pull out the drone brood, well, if you allow those drones to start to emerge, now you've basically given the mites everything they ever dreamed of to increase their population. You're going to have more of a mite problem than when you started. So it's a technique that works, but it's one that requires really strict commitment to the timing. The other good management technique that doesn't involve chemicals that can help to control mites is interrupting the bee brood rearing cycle. And so there's a few different ways that you can do this. One of them is splitting your colony. If you split your colony, depending upon the way that you do it, you can wind up with completely broodless colonies. You can wind up with colonies that don't have queens for a period of time. And in both cases, you've then created a brood break, a period of time when the colony doesn't have brood of a certain age. And all of the mites that were trying to reproduce are now going to be running around, unable to find anywhere to go. Maybe they'll die of old age. Maybe they'll make a clumsy mistake and the bees will grab them and chew their legs off. But something is going to happen that's going to reduce that mite's opportunity to reproduce. And so this can be really helpful as your colonies are regrowing. You can also use certain treatments while you're doing these splits if the colonies don't have supers in place, because some things can't be used with supers in place. There are other methods that folks use. So if you don't want to increase the number of bees in your apiary, but you do want to cause that brood break, well, one method is you can just let the bees swarm. Not necessarily what I'd recommend, but if your bees swarm, there's also going to be a brood break while they make that new queen. If you don't want the colonies to fly off into the trees, and you don't want to have more and more and more colonies each year, you can also cage your queen. And there are different protocols and methods that you can use to cage the queen and engineer a brood break, which is then going to allow you to manage the brood rearing in the colony in a way that will harm the varroa mites. So this is a little image that I created based on one technique that involves putting a frame and the queen into one of these queen cages, these cages that are made of queen excluder that hold the queen in place on a single frame while other bees are coming and going. And the basic idea is you lock the queen up for about 2 weeks, and then you let her out. And there's going to be a sweet spot, this perfect period, where you are able to remove the frame that she had been laying on, which is going to be riddled with varroa, and hit that colony with a treatment if you want to, and really, really knock your mite levels down. Now, the problem, of course, is that all of this math is kind of the simple version, which assumes that drone bees don't exist because they're capped for longer. So if drones do exist, now you can see the figure got a little more complicated. I'm not going to try to explain all of this right now, but I will say if you're interested in this, if you want to learn more, if you just Google Betterbee cage Varroa, the article that I wrote is going to pop right up, and then you can go and read it and learn more about this method. And I don't want this to feel like a commercial. You don't have to buy a cage from Betterbee if you don't want to. I just want you to understand that method if you're interested in it. So those techniques, those management techniques, especially when they're combined with miticides, are also weapons in your arsenal. They're ways that you can keep mites under control. If you don't understand how they work, something like drone comb trapping can really go sideways for you. But if you do understand how they work, you can sometimes find opportunities for synergy, complementary use of maybe a miticide and a management technique, or two management techniques, or two miticides. Combining and understanding how these things fit together can actually really supercharge your mite control. And again, waiting for just one silver bullet management technique is probably not going to be the best strategy. So the last topic I want to cover here is the idea of bee resistance to Varroa. And we know that there are a lot of programs all over the world that have been spending decades at this point trying to select for Varroa-resistant bees. There's a lot of different terms that you might encounter, and if I had another hour, I could go through each of these in some detail. But essentially, you might hear people talking about hygienic bees, bees that detect problems with the brood and then rip out the problematic brood, and that means they also rip out mites as they're reproducing. You might have VSH, or Varroa-sensitive hygienic bees. Those are bees that are particularly sensitive to the reproduction of mites in their brood. And the Harbo assay is just a method that people will use to try to score the VSH levels of their bees. You might hear folks talking about mite biter bees or grooming behaviors. The Purdue ankle biters is one line of bees that have been bred specifically to try to increase their grooming behaviors. The UBO is a tool that it's a product, UBO, but it stands for UBO, unhealthy brood odor. And it also takes advantage of some of these same cues that hygienic bees use to figure out if there's something wrong with the brood underneath. The pole line is a line that was created by the USDA, primarily the Baton Rouge lab, working with commercial pollinators and VSH breeding programs and trying to create a more mite-resistant bee. I've got some pole line genetics right out here in my home apiary that I've been playing with for the last few years. Russian bees are bees imported from Russia, putative mite resistance traits that have been selected for over time, and also survivor bees, getting bees that have survived with varroa mites under various circumstances, and then breeding from them, figuring that they've got some tricks up their sleeve that you might want to have in your apiary. So all of these tools are great. All of these traits increase the ability of your bees to fight against the Varroa mites on their own. And ultimately, what we want is bees that are so Varroa resistant that we don't have to do any other Varroa management. But at this point, in my estimation, we do not yet have a mite-proof honeybee. And that means that these partially mite-resistant bees really are an important foundational block in your mite control, but the management techniques and miticides are also still important. And some combination of these different things is going to be important to keep your bees from succumbing to the effects of these mites and the viruses that they transmit. So again, they can all interact with each other in either conflicting or synergizing ways. And we can't just wait for one silver bullet. We can't say, I bought Russian bees, therefore I'll do nothing else. You can't say, I split my bees every year, therefore I'll do nothing else. Because remember, the goal is not just getting your mite level low, it's keeping your mite level low throughout the year to avoid that virus accumulation. And so this mite management mindset, this mite management philosophy, you can think of sort of like putting all of these different tools we've talked about on a spectrum, right? Where on the left, maybe we're the ostrich with the head in the stand, we're doing less. And over on the right, we're going to grab every chemical out in the barn, right? Every pesticide we can think of, we'll throw into that hive, and we'll kill those mites. You probably kill all your bees too. Neither of those is a great strategy. So I want to tell you a story about some work that, or rather about something that I learned while I was doing some work on Varroa resistance traits of the bees in Madagascar. So that's me on the left, and this is Husman on the right, who was one of the technicians that I worked with. And he's using a paper clip that we hammered into a grafting tool because we couldn't get any grafting tools in Madagascar. So we're trying to breed these mite-resistant bees, and we're going out to these villages where these folks have been keeping bees, and with varying success. And this is one of the villages where we went out. We were teaching them how to use a refractometer to measure the honey levels in their hive. And as you see, this village is largely wood harvested from the local jungle. There's some sheet metal that's brought in that is used for roofs, but there isn't a lot in this area that isn't grown or produced in the local environment. But something that I noticed that was weird was as I was walking around, there were all sorts of these medical blister packs, things that had previously held pills. And I said, what are those? And they said, oh, that's what amoxicillin comes in. You can go down to any little roadside stand and somebody will sell you some amoxicillin. And I said, why? Are there really bad infections that they need to use this antibiotic? And they said, oh, no. In Madagascar, if you have a headache, if you don't want to do anything, you can just kind of grin and bear it. But if you want to treat that headache, then you want to buy medicine. And antibiotics are the strongest medicine that all of these villagers have ever encountered. They've seen people at death's door turn around when they take antibiotics. And so if they've got a really bad headache, they're going to pop an antibiotic because that's definitely going to be the thing that helps them. Obviously, we know that the middle ground, taking an aspirin, is a better idea than popping antibiotics when we just have a headache. But that mindset, I think, sometimes it trickles into the way beekeepers manage their Varroa mites as well. And so if we look back at this Varroa management spectrum, as I said, on the left, we're the ostrich with the head in the sand. We're just pretending that Varroa don't exist and hoping that nothing goes wrong. On the other end, though, let's say we're going to rely heavily on those synthetic miticide products, particularly Apivar, because that's the one the mites haven't developed resistance to. Well, we've also got other products, the organic acid-based products and the essential oil-based products, the thymol products, and the oxalic acid products, things like that might be a really helpful way to keep mites under control. We've got physical intervention, like splitting our colonies, caging our queens, or doing drone comb brood removal. And you've also got resistance. This is a map that was taken from a research paper talking about all the different places around the world where resistant honeybees have been identified. And if you're squinting, trying to figure out what that place in North America is, it's the Arnott Forest, which is where I did my doctoral work working with Tom Seeley on the resistant or the partially mite-resistant population of bees that live in that forest. So combinations of these resistance traits, physical interventions, essential oils, organic acids, synthetic pesticides, all of that together can be used to control Varroa. And so if you are a beekeeper who doesn't want to put chemicals into your hive, you might want to draw a line on the spectrum and say, I'm going to do the stuff on the left, not the stuff on the right. And that's an approach that can work, although often I have seen it go more wrong than right. However, you might also draw a line like this and say, I don't want to use synthetic pesticides in my hives, but I'm comfortable with essential oils. I'm comfortable with organic acids. So where we are in this spectrum can give us a sense of how we are going to be mixing and matching different tools. Now, where on earth should we put the brand new genetic technology, RNAi technology? I have no idea. Is that, would you put that up with the synthetics or would you put that down with the essential oils? I don't know. But the important thing is that we can create this sense of products that or tools or techniques or genetics that I might use and things that I might not, and then create a Varroa management plan. I've used that spectrum to sort of illustrate my point, but really the way we should think about this is in a pyramid shape, where at the foundation of that pyramid, we have cultural control, bees that are as resistant to mites as we can get, maybe brood breaks that we engineer into the colony by splitting or allowing swarming. Then we've got mechanical control, dumping powdered sugar on your bees. Yeah, it'll kill a couple of mites here and there. Trapping mites in drone comb, that's a mechanical control. And then as we move further up, you've got chemical controls, but we can divide that into soft chemicals, organic acids and essential oils, and harder chemicals, synthetic pesticides. That's not to say that we aren't going to use the synthetic pesticides. Just like in the food pyramid, it doesn't say that you're never going to eat candy, but you don't want candy to be the foundation of your diet or you're going to have a pretty hard time. So as a beekeeper, I recommend creating a Varroa management plan. And in order to do that, you need to answer these questions. Then none of them should be terribly hard. How often am I going to inspect my hives? Will I test for mites? And how often will I test for them? How will I test for mites? Am I going to use a sticky board or am I going to use the alcohol wash? Will I treat all of the colonies in an apiary if one of them tests above the threshold? That's a decision that you can make. What non-chemical mite controlled methods am I going to be using in my hive? And what chemical mite controls am I comfortable using and do I know how to use? If you can answer these questions, suddenly it becomes a lot easier looking at this upcoming year to make a good plan for Varroa management. This is an illustration that I've pulled from the last few years of Betterbee catalogs, where we always try to include Varroa management sample plan, just to give people a sense of how you might fit some of these products and ideas together. And so what I want to emphasize is that none of these plans, 1, 2, or 3, is the plan that I recommend to every beekeeper, right? There are products that aren't listed. There are circumstances under which you may or may not want to do exactly what's written here. But essentially, if you had absolutely no clue what to do with Varroa, you could stick yourself under plan 1, plan 2, or plan 3, and roughly every month, check your colony, figure out if they need intervention, and then intervene. For the most part, this is going to list miticides, but there are other techniques that you can use. Things like drone comb trapping could be combined with some of these other products. And so making a plan is I think, the most important way to ensure success of your colonies. And testing the colonies periodically is the best way to make sure that the plan you made is working. Because you can't make a plan, do a bunch of things, never measure your mite levels, and then say, well, it can't have been mites that killed my bees because I did a lot of things. Well, if you don't confirm that what you're doing works, you haven't really made a functional plan. With that, I'd like to thank you for your time. Trained by academia, I've spoken for exactly 60 minutes. And so if anybody needs to go, you're more than welcome to do so, but I would be very happy to answer questions. If you do have a question, but you don't have the opportunity to ask it or get an answer right now, you're always welcome to send an email to support@betterbee.com, and then that will get directed to me. You can just say, hey, I have a question for David. Can you pass it over to him? And they will make sure that I answer you. So thank you very much. I will stop sharing my screen now, and we can take a look at the chat and see what's been going on. And I'm happy to chat with you folks for as long as you've got questions for me. But first, I'm going to take a sip of water because I've been speaking continuously for an hour. Okay. hearing no other plan, I can just start at the top of these chat questions and see where that gets us. So Anthony has asked, is there a good way to determine whether there are any feral bee colonies near you? Oh, that's a good question. Since they're untreated, it would appear that they would fall into the bad neighbor category, being a source for Varroa and possibly other issues. It's a marvelous question. So one of the best ways to try to figure out if there are bee colonies near you, I don't know if they're feral or if they're your neighbor's bees, is putting up swarm traps. If you put up a bait hive, which is the term I prefer to use, a nice, attractive box, put up at some elevation and then, presented in a way that scouts are likely to find. If you are catching swarms and those swarms are not coming out of your hives, you have now determined that there are bees within flight range of your hives that could be interacting with your bees, in a disease ecology way, spreading mites to them or picking mites up from them or what have you. So that is a good technique. There's obviously beelining is a fun hobby. I think I have Tom Seeley's book right here next to me. Following what did he call it? Following the Wild Bees. There it is. There you go. So if anybody would like to pick up a little bit of light reading, this is a thorough explanation of how one would set up a beelining box and go out and start hunting the forest for feral colonies. I can tell you, having looked for feral colonies, I spent hours wandering around the forest looking at the same tree and not finding the bee colony that lived inside of it, and then suddenly seeing, oh, that's where they're coming and going from. So it's not an efficient use of one's time. In many ways, I think setting up swarm traps, and if you don't catch any swarms and your bees don't swarm, that seems to be the most reliable way to say, yeah, I don't have too many neighbors. Another question, where does Apivar 2.0 fall regarding mite resistance with it? So Apivar 2.0 still uses amitraz as the active ingredient. I have seen some little glimmers in some research studies with the new formulation suggesting that maybe it is very, very marginally more effective against mites that have low levels of amitraz resistance. But essentially, no. If your mites are resistant to the active ingredient is not going to kill them. So the best research that I've seen tells us that if you have amitraz resistant mites, if those products aren't working, you need to go on an amitraz diet and use other products to control mites for a while, and then you can try amitraz again and see if it has regained effectiveness as the mites that are resistant have been sort of outcompeted and out-reproduced by the mites that are susceptible to it. Another question that talks about Varroa. So this is just a link to a podcast episode, the Beekeeper's Corner podcast, that discusses Varroa. So folks can click on that link or bookmark it for another evening of mite learning if you want to do that. And Robin Underwood from Penn State has mentioned that Anthony's the only person typing in here. Robin Underwood mentioned that Canada has recently approved letting beekeepers make extended release oxalic acid strips. What is the chance of that happening in the US? So I am in a monthly video call with a lot of the people in Canada that made that happen. And that includes also folks who work for the federal EPA in the United States. And unfortunately, the way our rules are written, it seems unlikely that we are going to be able to do that. I will say I am also working with a miticide company right now on a product that would allow beekeepers, it's sort of a half-made strip, and then if you buy that product, you can finish making it, and that might be a cheaper and efficient way for beekeepers to make up their own strips. But realistically, the way U.S. pesticide laws are written, there are things that are easy and there are things that are hard. You, beekeepers of Vermont, managed to attempt something that no one else had attempted to see if there was a backdoor that we could get through. And the answer is, unfortunately, no. That got slammed in our faces. And so we're not going to be able to make our own strips, at least in the foreseeable future. Where does queen breeding for Varroa-resistant traits fall into my scheme of mite management? So I think I discussed that already, but the question, and then there's also a question about whether Saskatraz bees exist. They do. And often what you have is that there will be a breeding program producing a particular bee, and then whoever's running that program will stop, but maybe they'll give those bees or sell those bees to someone else, and now they're going to put a new brand name on it, or they're going to mix in some other genetics from somewhere else. And so it can become difficult to keep track of what breeding program your bees are even coming from, or which bees are or are not available. And so the short answer is there are a lot of really diligent queen breeders doing a very good job taking bees that are partially mite resistant and making them more mite resistant over time. The issue is there a bee that can be kept sustainably without any mite treatment? Yes, there are people who are consistently not treating their bees for Varroa and are able to keep those bees alive. Can I take their queens and can I put them into my beekeeping operation? And can I manage them the way I want to manage my bees to maybe minimize swarming, maximize honey production, and not manage them the way that the queen breeder is using, and still have them be completely, resistant to Varroa? I have not seen evidence yet that that bee exists. That's the goal, right? A bee that's just so Varroa resistant that no matter how we keep them, we don't have to think about these mites. But unfortunately, what we tend to see is that very, very good bee breeding work makes bees that are incrementally more mite resistant and therefore probably need less intervention. The mites are going to get less out of control, at the same speed, but they still often require some kind of intervention, be that chemical or non-chemical. Oh, and here's another question. So this made reference to the seminar that I just gave down at the lecture I just gave at the Saba seminar on Saturday. And I asked two questions. So this is really veering into a slightly different topic, but I asked two questions there that I've been asked to repeat to you. And so I make everybody turn to their neighbor and speak in the room and sort of come up with an answer, but I won't do that to all of you here. I'll just ask you to sort of sit and ponder for a moment to yourself the answers to these two questions. Question one is, should we, as beekeepers, be allowed to violate pesticide laws if doing so, we think, is going to allow us to keep our bees healthier? Right? Should we be allowed to violate the laws and rules that regulate these miticides if doing so is going to make, our colonies live longer, is going to help us kill Varroa mites? And then I make everybody sit and think about that. So I want you to just ponder, what's your answer? Deep in your heart, what do you think the answer is? If I'm worried my bees are going to die if I don't break the rules, should I be allowed to break these pesticide laws? That's question one. Question two, after half the people in the room have raised their hands and said, yeah, we should be allowed to break the rules, I say, what if instead of being a bunch of beekeepers, we were a meeting of a whole bunch of apple growers or blueberry growers or whatever the case may be? And the question is the same. Should we, as apple growers, be allowed to violate pesticide laws if we think that by doing so, we can increase the success or the health of our crop? As beekeepers, what do we want the blueberry growers and the apple growers to say? We want them to say, hell no. We've got to follow every pesticide law exactly the way that it's written. Because if we don't, those honeybees that we pay to bring in and pollinate our crops are all going to die. And so as beekeepers, we generally benefit from a mindset that says every single pesticide law should be followed exactly to the letter, and the pesticide regulators should enforce those laws with bazookas. They should find somebody breaking the law and they should blast them into orbit. Because that's the kind of mindset that's going to keep our bees from being exposed to harmful pesticides. If instead we get into this relativism where we say, well, obviously we can't do this and we can't do that because Varroa is so hard, so we should be able to do whatever we want under every circumstance, it makes it extraordinarily hard for us to go and actually ask beekeepers or rather ask growers to follow the pesticide rules because they know that we're the hypocrites who are not following them ourselves. And so this is a point that has taken place, that beekeepers that have gone and tried to lobby for things like neonicotinoid bans have had thrown right back in their face the fact that beekeepers are famously ignoring some of these pesticide laws. And that really undermines our ability to get anything done to protect our bees. And so that's why it's so critically important that if you are using miticides, and I do recommend that most beekeepers use miticides for part of their mite management, that we're following the laws, we're doing what's legal, we're using the correct doses. And if we don't like something, then we have to push against the laws. We don't just ignore them and say, what's the worst that could happen? Because the worst that could happen is that we aren't taken seriously. Okay. And then the last question is, are these slides available? So I don't actually know exactly what the plan is for the recording. There is a recording that has been made of this meeting. So Brooke, you can comment on under what circumstance this will be available to folks in Vermont.
Yeah, we are going to post it on the apiary page of our website, the Vermont Agency of Agriculture website. So
Yep.
They won't be the slide deck per se, but I think the recording is better than just having a slide deck without
Right. You'll have to listen to me rambling over my own slides, but you'll be able to see all of this stuff again.
which has been wonderful. So yeah, I really appreciate it.
Right. All right. Well, Anthony's mentioned a few sort of crackpot methods that do not have good evidence behind them, or at least don't have enough evidence behind them yet to be viable products. Looks like no other questions. If there are any, by all means, throw them in, and I can answer. But otherwise, I will bid you all a good evening and go refill my water cup.
Wonderful. Thank you again, David. It's been great and an honor to have you. I learned a lot.
Great. I'm glad. It's always a pleasure. It's always a challenge to, if you've got a brand new beekeeper and a beekeeper who's been keeping bees longer than I've been alive, it's a little hard to know how to present this. But my goal is to teach everybody at least something.
Yeah, I think it worked. I mean, I feel like I know a lot, but I also learned a lot too. So, and the new miticides coming down the pike is really exciting.
Cool. Yeah, any new tool, any new weapon in the arsenal makes us better able to keep our bees healthy.
Yes.
All right.
Well, excellent.
Thank you so much.
Thank you so much. I'm gonna end that.